everyone. So I'm based at Kings Park. I run the orchid conservation program there. So you're going to hear a lot of orchid heavy content. Maybe that's good or bad. But really what I'm talking about are the interactions that orchids have with their environment. So think about the species you work with while I'm talking about orchids and how the interactions are often very much the same. There we go. So if orchids were people, they would 100% know how to win friends and influence others. They really are networking specialists, and that happens both above ground. So they have really highly co-evolved floral morphology with their pollinators. So there's chemical, physical and nectar cues for pollination to happen. And also below ground. So orchids have obligate reliance on a mycorrhizal partner for germination, and then depending on the species and more so where you are in the world, they can need that re-establishment each year, so particularly temperate orchids. It can be a very specific interaction or it can be quite generalist, but it is the orchid's main way of getting nutrients out of the soil, so it's very important to them. Orchids also exploit niches throughout their whole life cycle. So during reproduction, they produce vast numbers of seed, probably in part because not that many of those seed actually make it to adults. But they have a very high dispersal capability, and the plants themselves can be quite long-lived. They also exploit niches in their nutrition. I mentioned they're fungi. Orchids don't really have root systems, so those fungi are replacing that root system, and so they're able to get to nutrients in the soil that a lot of the co-occurring shrubs and trees can't actually get to. And in their life history, they snooze through the hardest parts of the year in temperate environments, so they go dormant for summer and they ride out fire and drought, and then they're back up for when it's rainy and a lovely time to be about. So they've really cornered the market on how to make it okay for themselves. So when we're talking about orchid translocations, we're not just talking about putting a plant back in the ground. We're talking about making sure that all of those interactions are present at a site. So it's finding the right habitat, it's making sure their fungi are there, but it's also making sure that the habitat is right for the fungus as well, so maintaining that symbiosis long term. And in the pollinator side of things, it's making sure the pollinator is present, but it's also, sometimes our pollinators have really tricky life cycles. So a lot of orchids in WA are pollinated by a thinid wasp that parasitises a scarab beetle. So you need to make sure that habitat supports scarab beetles as well as the wasp and the plant and the fungi, and when they all come together you get this very narrow band right in the middle, and that's what we're looking at trying to replicate or reproduce. So when I talk about translocations in public talks, everybody thinks of the picture on the far right, planting day, which is a very exciting day, it is. But there's a lot that goes into getting to that day, and some of those things are slightly different for orchid translocations. So as with everybody, we are looking for seeking approvals maybe a year before. Site selection is sort of 1 to 2 years before, but maybe where it starts to differ when we're looking at interactions is that before we've selected sites, we want to know that the pollinator is present. So there's quite a bit of research, and it can be a few years of finding out where those pollinators are and how we can best support them. Happening at sort of the same time, it overlaps a bit depending on the species, is looking into propagation. So finding the fungi. Do we have the right fungi? What is the right fungi? Can we grow the plant? Do we hold it back in the glass house to develop that tuber resource so that we know that they'll have better survival. And then the very first step, Andrew touched on before, starting early. So some of our ex situ collections are over 20 years old. That's really the first place to start with any, well, I'd say conservation in general, but translocations in particular, is making sure you've got that material to begin with. When we look at post-planting, so this is borrowed from the translocation guidelines and I guess slightly tailored towards orchid translocations, keeping in mind those interactions. So in the short term, the success criteria we're looking for is just survival, but the key interaction that we would have had to have set up for that to happen is that mycorrhizal partnership. In the midterm, the 3 to 7 years, we're looking at reproductive function, so flowering, seed set is happening at site, and so that's where we've made sure that both mycorrhizal and pollinator interactions are happening at this site. And in the long term, like everyone, we're looking for self-sustainability and recruitment at a site, and when we've got that happening, we know that we've hit all three of our key interactions, which is habitat, mycorrhiza and pollinators. That's that middle of that Venn diagram, and that's what we're aiming for. So when we start to consider ecological interactions, our long-term success is really very much dependent on quite a significant, at times, pre-planting investment. So there's a growing body of literature around the world and in Australia on orchid translocations. And so we know now that the first step in translocating with interactions is to understand how to either reproduce, manage or mitigate threats to those interactions. We know that translocating with mycorrhiza, so symbiotic propagation, increases survival from 50 to 61%. And we know that making sure that pollinators are present increases fruit set from 25 to 63%. So these are really our two key interactions that we want. If we look at mycorrhizal interactions, again, Andrew mentioned ex situ collections. We're not just banking material, we're banking knowledge. When we screen our seed and fungal collections, we know that they are ready to go should we need to call on them into the future. So a good example of that is this little green orchid here. This is Pterostylis sinuata, or the Northampton greenhood. We have collections from the 90s for this species, but those initial germination efforts failed and it was sort of stored but put to the side as something we need to come back to. Recent collections, we were able to collect 17 isolates. It was a really good year. And in collecting so many extra isolates, we were able to find out that actually this species carries a lot of fungi that doesn't necessarily enact germination, but we've now got those 2 isolates that worked banked away, so we know that this species is ready for conservation efforts if we need to. Probably where we have a bit of a knowledge gap is in the long-term fungal longevity across genera. So we're just starting to get into orchid fungal physiology now, and we have a few questions around storage for some species. So in translocations with mycorrhiza, we've got good news because symbiotic propagation means that we can really jump that germination hurdle. So we can go from a natural germination in the wild of, say, 1% up to anywhere between 60 and 95% germination in the lab. We're also able to deliver that mycorrhiza to site, and that becomes an inoculation source as plants re-emerge each year. And so we also plant soil cores because that delivers that symbiosis intact to the site, so we can see better survival in our plants by doing this. So there's a few, I mentioned fungal physiology is probably a path forward into the future to better understand you know, how can we harness these fungi to do a better job for us and on behalf of the orchids that we're working with? And there's a few things to play around with in terms of planting density and also where we get our fungal friends from as well. So on to pollinator interactions. So we're really interested in understanding who is doing the pollination, how they're doing it, so what kind of pollination syndrome are the plants using, and one of the first bits of information we might have about a threatened species is how often is seed set occurring, and that can tell us an awful lot about what we might be facing when we're going towards translocation. So we're really keen to know if potential planting sites have pollinators present, but sometimes when we're working with really threatened species, that means that we have to wait to have flowers, so we have to grow the plants, have the flowers before we can start collecting this information, which is why I had that 3 to 7 year time frame. So an example is Bustle spider orchid. So Bustles is in the middle there, and those pictures just give you an idea of pollination syndromes. So on the left, sexual deception, so red labellum, clubbed, releasing pheromones, attracting wasps, and on the other side is something that's food deceptive, so very bright white, might attract quite a few different things. And the spectrum, the syndrome that your plant is using can have a great impact on how your translocation goes. So are you looking at making sure a site has one pollinator, or are you looking for a suite of them? And you may even be faced with the management commitment of having to hand pollinate flowers as well. So with Bustles, we had a lot of visitors, but we wanted to know who was important. So this was a three years of work and a really intensive set of surveys in 2022, which established that the wasp was the main pollinator, but we had some of these other visitors, and this wasp was at 3 sites that we were potentially planting into. So to give an idea, when wasps were present, we were getting seed set. That's that little green bit. Most of the pollen was staying in the flowers, so it was quite a good, removal to seed set happening. Where we didn't have wasps, we were getting a lot of pollen taken away, so we were losing 60% of the pollen out of the system and getting no seed set. So it can make a really big difference to get that pollinator interaction at your site. And so we've set up some of our translocations to look at planting density to understand how that might improve seed set as well. So we're not yet at restoring pollinators. It's a bit of a research gap, but we have touched on co-release a little bit today, and we could be looking at co-releasing the food plants of the wasp pollinators. So those blue bars on the graph are what's flowering at a site, and the orange bars are the pollen loads on the backs of the wasp. So lots of stuff in flower. Wasps are only going to a few key species, so we could be planting these out at the same time. A specialist interaction that probably doesn't get anywhere near enough airtime is human interactions. We're all specialists in our field here today, sharing ideas. We often use volunteers, connect with departmental staff, and have a huge number of funding partners. So I think it's really important to acknowledge a different kind of interaction in our work as well. So I've talked a lot about orchids, but beyond orchids, we can have a look at the species we work with, and as we start to understand the ecological interactions, we can really harness them and use them to our benefit and the benefit of the species that we're working on. So we know that 80% of flowering plants have a mycorrhizal association of some kind. Doesn't tend to be at the obligate end of the spectrum like orchids are, but we can certainly consider mycorrhizae in our other translocations, and along with our pollinators and potentially co-releasing pollinators into sites as well. So I'll just leave you maybe with a question that comes up occasionally when I'm talking about such intensive interactions, which is what level of management, intervention or research investment are we comfortable in deploying in the conservation of a species. So I don't expect answers, but it'll be different for everybody. It depends who you are, who you work for, what you're working on, how much funding you have, but it's really important to think about interactions in conservation as well. So I will leave it there.